Hello everyone, I'm delighted to be here and thank you for the organizers for inviting me to present this presentation. Which one is it? This one. These are my disclosures and interests and funding as well as IP. Then just a few of my collaborators. There are many, many more all over the globe, but this is just my close collaborators, Professor Douglas Kell, Amy Proel from Polybio, many, many more, and also my team of research collaborators and students and workers in Stellenbosch University. Then just briefly my research focus. Over the few past years, I have been looking at inflammatory molecules that may cause pathological blood clotting, including those coming from both viruses and bacteria. And we have been interested in the effects of circulating inflammatory molecules on platelets, on red blood cells, as well as the clotting protein fibrinogen, which is the main clotting protein. We are also interested in development of novel methods to study clotting pathologies, and these include imaging flow cytometry, proteomics, and also fluorescent microscopy. Over the years, we've published many publications looking at these inflammatory molecules and their effect on the blood system and cells in the circulation. Then came along COVID. So, just a brief background for those of you that might not perhaps know the circulating inflammatory molecule interactions with platelets. So platelets are very well known that they have numerous receptors on their membranes that can interact with each other, the platelets themselves with each other. They can interact with all of these inflammatory molecules via these receptors. They also play an important role in interaction with the endothelial layers, especially when there are damaged endothelial cells. So platelets are known to form complexes, not only with themselves, but also with other cells of the immune system in circulation. Pathological clotting, as I mentioned, is also one of the things that we study. Of importance is how proteins that are of inflammatory nature might interact directly with the protein fibrinogen. So if you look at the protein fibrinogen, it's usually supposed to be in a soluble form when you are healthy, and it changes into fibrin when there is a clotting necessity. However, in circulation and in the presence of inflammatory molecules like the spike protein, these molecules can directly bind to the fibrinogen molecule and change the structure. And as early as 2016 already, myself and my collaborator Douglas Kell showed that this changes due to binding of inflammatory molecules might cause the protein to change its structure into an amyloid shape. So we're not talking here about amyloid that we know in the brain of Alzheimer's and Parkinson's. It's the amyloid protein change in the blood, in the fibrinogen itself, and we have marked this with various fluorescent markers that bind specifically only to amyloid, and one of the markers that we use is theoflavin T, a well-known marker to identify amyloid protein. So just to return to COVID and long COVID. So we need to look at acute COVID to understand the clotting pathologies and the thrombotic endotheliitis that will result in long COVID. So if we look at what happens in acute COVID and then also translated to long COVID, there are numerous activation processes happening, particularly looking at the platelets as well then as fibrinogen, where there are these activation processes that activate and damage the endothelial layers. We know that from acute phase, but we also know that is happening in long COVID. So early on in the pandemic, we took blood samples when it was when everyone was in lockdown, and we studied and looked at the platelets, and to our surprise, we found numerous platelet hyperactivation and damage in acute COVID. Here you could see structural changes in platelets with the yellow arrows, where the platelets looked as if they've just exploded, and also interactions with red blood cells. And then we also found microclots. We did in that stage, 2020, did not name it microclots, microclots. We only did that when we started looking at long COVID. And here you can see with the blue arrows where these microclots are just packed onto red blood cells and also glue together the red blood cells. So you could just imagine that in acute COVID, these samples looked very much sticky, and our reports from our clinical collaborators globally also mentioned the sticky blood in acute COVID. Then I mentioned previously we looked at studying the amyloid's nature of fibrinogen. So we also then obviously took the chance to look at this in our acute COVID samples. Here's just an example of healthy plasma, type 2 diabetes, and then of COVID plasma. So we simply added theoflavin T, our marker for amyloid, to platelet-poor plasma. We exposed it when it was freshly drawn from the patient. We exposed it, waited for 30 minutes, and put it into a fluorescent microscope. And here you can see even healthy plasma people who are healthy do have small abnormal areas in their fibrinogen. We see that in every single healthy individual. However, it's the size and the nature of these clots that might differ. Type 2 diabetes, we know they have inflammatory nature, and we also know many people suffering from type 2 diabetes have got a higher propensity to clot and form clots. We know deep vein thrombosis is very well known in this disease. To my surprise, in acute COVID, I could not believe what I was seeing in front of my eyes. The amount of these amyloid structures in the platelet-poor plasma were significant. It just lit up like Christmas trees. And then from that moment, we knew that there was something that we need to look into further. We also looked at the platelets, and just to just make sure that we are all on the same page, these platelets are exposed to two markers, CD62P, which is the Pinckney signal, that's for P-selectin, a well-known platelet marker, and then PAC1, which is a glycoprotein marker that actually indicates that it is platelets that we are looking at. These platelets were also significantly hyperactivated. You could see a typical healthy platelet in a healthy individual, and from B to F, you could see the platelets from acute COVID. Because we were working with a fluorescent microscopy method, we decided that we need somehow to assist clinicians, not perhaps looking at research in such detail, to understand what we are looking at, because obviously we know microscopy is not the ideal type of method to try to look at quantifying damage. So we developed a platelet grading system where we said that one is when you have a healthy individual with little spreading, and in a healthy individual there should not be any clumping, so no clumping. And then 2, 3, and 4 is progressively the platelet damage that one would expect. Perhaps 2 you would find for the spreading in a healthy individual, 3 perhaps in a typical diabetes individual or someone with rheumatoid arthritis, and then 4 would be where we would find significant platelet hyperactivation in COVID. We also developed a microclot grading system based on the same type of criteria, where 1 and perhaps 2 might be in a healthy individual. 2, the grading system for 2 is perhaps someone that's older, perhaps a little bit more inflammation, and then 3, 4, and 5 were the huge microclots that we typically would see in a COVID population. So then we turned our attention, as most of us did, to long COVID. So very early on in the end of 2020, beginning of 2021, we had our first long COVID reports in South Africa. And we also looked at their platelets. There you can also see platelets still very much hyperactivated in long COVID patients that were at that stage. There wasn't even in South Africa a proper belief that there was something like long COVID. People weren't even recognized that it could happen. This is just an extremely badly activated micrograph of platelets in long COVID from someone who suffered from this disease for 3 years, still having significant platelet hyperactivation. We also used our scanning electron microscope analysis methods again to closely look at how platelets would look like. Here you could see on the left a healthy platelet, and on the right just massive platelet clumping together, breaking up, and absolutely not looking very healthy at all. And then once again, also in long COVID, we did again, as with acute COVID, found significant microclot formation. Here you could see how the red blood cells are just packed with clotting protein. And one can imagine that those red blood cells would not be able to fulfill their duties as they should by delivering oxygen to the organ systems. And one can just think that they can also clog these little red blood cell clump themselves, can clog up the finer capillaries, because they will definitely not be able to flow significantly well through the blood vessels. This is just a close-up of the red blood cell of a long COVID patient with the microclots just packed onto the membrane. And once again, if one look at this scanning electron microscope view of a red blood cell, one can imagine that the red blood cells would not be able to function properly when they are so severely impaired with microclots covering their periphery. Microclots in whole blood, we also looked at that again, once again in long COVID, and here you could see these clots it was simply just whole blood, a drop placed on a glass slide and prepared for scanning electron microscopy, nothing added. So this is straight from the patient, minutes after the patient had given the blood sample. So the questions that we sometimes get is, these microclots, are they also present in other diseases, and why haven't we seen them? Yes, they are. We have been showing that microclots are present in lupus samples, in obviously acute COVID, in rheumatoid arthritis, also in conditions like Alzheimer's and Parkinson's, in all diseases where there are clotting pathologies or coagulation pathologies noted, and all of these are well known for that. We also looked at the spike protein. In 2021, we added spike protein, the S1 part of the spike protein, to healthy blood, and we could induce, just by adding the spike protein itself, the S part, to whole blood and to platelet-poor plasma, we could induce significant platelet hyperactivation as well as induce microclot formation. So we do now, from that publication, we hypothesize that spike protein can actually have direct protein-protein interactions with fibrinogen plasma proteins as well as with the receptors on platelets. And here's just some other work from that publication where you could see how the platelets were just significantly clumped together, hyperactivated, binding to red blood cells was seen. And we also did a microfluidics analysis where we compared healthy plasma at the top, you could see it on the right, a COVID example where the clots flow through, and then the top sample exposed to spike protein at the bottom, we could induce significant microclot formation. With a team of US collaborators, we also looked at Omicron and compared that to the beta and the alpha, the older, more virulent variants, and we did recognize that in Omicron, the clotting pathologies were not as significant as we saw with the earlier variants. Interestingly, the platelets in Omicron samples were still significantly hyperactivated, and we could not see the difference between Omicron and the more virulent variants. And this is just an example of just some of our unpublished work that wasn't included in the paper, where we looked at the platelets from Omicron patients. Now, we also turned our attention to proteomics. So proteomics, we used MS, mass spectrometry, and we followed the usual processes where one would digest the sample, the protein sample, with a single trypsin digestion step. Unfortunately, we could not digest all the plasma clots in the acute phase as well as the long COVID samples. We could not digest our protein with a single trypsin digestion step. We had then developed a second trypsin digestion step, and only then did we successfully digest the clots. And we looked at, after the first digestion step, we looked at the microclots, and we still saw that in acute COVID and in long COVID, we did not digest the clots. However, we also compared it to healthy and to type 2 diabetes. We indeed could digest the plasma with a single trypsin digestion step with in the controls in the diabetes, but not in the acute and the long COVID. The second step, we could digest everything. And then when we did our proteomics analysis, we were surprised to find, or perhaps not so surprised, I think, we found numerous inflammatory molecules, von Willebrand factor, C-reactive protein, some of the complements, plasminogen, fibrinogen, obviously, because we're looking at the clots, but also a molecule called alpha-2 antiplasmin. That immediately drew our attention. And if you look at the clotting cascade here, which I'm not going to go into detail, but just focusing on where alpha-2 antiplasmin have got a function, it actually blocks clot breakdown. So now we had a reason why we see the clots in circulation that are not breaking down. In 2022, we did a further analysis, proteomics analysis, with a much larger sample. This is also what we again found. All the molecules in the first experiment were there. What we also now found was numerous antibodies entrapped inside the microclots. We published it, and we're not a immunological group, and we did send it to some of our collaborators, and we still do not know exactly what all the immune antibodies that we have been finding in the clots, what they actually mean. So it would be great to study that further. So now the question arose, is there another way that we could try to give clinicians some assistance, pathology labs, how to identify these microclots in circulation? And luckily there were. We found a place for flow cytometry, in specific imaging flow cytometry. So in the beginning of the year, we received a crowd-funded and supported by Polybio as well as Balvi Foundation. We received our imaging flow cytometer, and we developed a method to look at microclots in a cell-free method in cytometry, with flow cytometry. We know that flow cytometry is usually used for looking at cells, and we removed the cells, we used platelet-poor plasma, and we could indeed find significant differences between our controls and our long COVID. We used our marker for it, theoflavin T, and we are in the process of looking at other markers, endothelial markers, other amyloid markers, and also biofilm markers in using flow cytometry to better quantify and classify the microclots. And here you can see some data from our recently published paper on our method. Now, we also looked at other possibilities, automated systems for microscopy. We are busy with a meta-systems automated analysis for platelets, which turned out to be quite difficult because these clumps are quite huge. The platelet clumps are quite huge, but we could find a method. We developed a method. We're still working with meta-systems also to try to see how we can further work on the Z-stack and to get all of the information. And we're also busy looking at the meta-systems automated microscopy system for microclots. However, the flow cytometry, I think, is a much better method because many pathology labs have got that. So let's quickly look at the bigger picture in the last few slides of microclot presence. What do we know? We do know that they are widespread in all chronic inflammatory diseases. We have found them ourselves in Alzheimer's, Parkinson's, rheumatoid arthritis, and many more. We do know that they can be stained with fluorogenic dyes, such as theoflavin T and also the amitractor dyes. And we do know that they are resistant to the normal process of fibrinolysis. And ourselves have found alpha-2 antiplasmin as possibly one of the mechanisms that prevent clot breakdown. And we do know that we can actually induce them in vitro by adding spike protein to healthy blood. And we do know that we can also induce other kinds of microclots by adding bacterial lipopolysaccharides, LPSs, lipoteichoic acids, even estrogen and other inflammatory molecules. If we add that to whole blood, we can induce other forms of these microclots. And interestingly, and that's why I say other forms of microclots, interestingly, we believe that these microclots can exhibit considerable structural and even spectral heterogeneity, reflecting the molecules that were bound to them in the first place. So a variety of possible changes might happen due to the polymerization process. So microclots and endothelial damage, we think point to heterogeneous and complex disease pathology. It is not simply a size or numbers of the microclots present in healthy and long COVID or any other disease, but it's also their content, their activity, and the biochemical characteristics that we must look at. And we cannot forget the activated, hyperactivated platelets. Ultimately, we believe that these two pathologies drive thrombotic endotheliitis in long COVID. The next steps in my last slide, we are still continuing research with all interested collaborators all over the world, so I extend an open door to you here. If you want to join us in finding answers, please do. We have got commercial endeavors because we do believe that the imaging flow cytometry and microscopy need to get to the patient where it's needed the most. We are determining ranges of microclot presence. We are doing a third proteomics analysis to identify more markers, and hopefully there will be some clinical trials looking at anticoagulation in the US as well as the UK, and then we are busy with numerous grant applications. And then I just want to, before I end off, just remind you, perhaps you have seen it, there's a wonderful piece of research out from Scottish collaborators that show that they followed 1.1 million COVID patients over a few period of months, years, and they determined that the only thing that prevented these patients from going into long COVID after they had acute COVID was the usage of anticoagulants. So we do believe there's a place for anticoagulants to treat at least the endothelial pathology in long COVID. Thank you.